Hello everyone, welcome to our third video of our Codeon AP Biology series. Today we're going to be learning about the cell. So just a little background, scientists have something today called the theory of endosymbiosis, in which they believe that the organelles in eukaryotic cells, so more complex cells, were once free-living prokaryotes, so single-celled organisms with their own DNA. And how they know this is because two organelles, chloroplasts and mitochondria, in these eukaryotic cells have their own DNA. So this is something that we'll talk about more later. And we all know that cells abide by something called the modern cell theory, in which cells come from pre-existing cells. That's the first condition. Second condition, cells are the smallest unit of life. And then third, all living things are comprised of cells. And these cells have common characteristics. They all have plasma membranes, components of their cytosol, and also they have DNA, so the instructional code, and also ribosomes to assist in the synthesis of proteins. Now, as I mentioned before, there are two types of cells. There's prokaryotic cells and eukaryotic cells. So we know that prokaryotic cells are very simple, they're small, and they're unicellular. And what differentiates them from eukaryotic cells are that they don't have a nucleus or an internal membrane. On the other hand, eukaryotic cells have a nucleus, and they also have organelles, membrane-bound organelles. This allows for more efficient processes, like the synthesis and packaging of different proteins, and eukaryotic cells are also a lot larger than prokaryotic cells. So we can first begin by talking a little bit about the structure and the function of cells. So we know that there's this very important theme in AP biology that you must know, is that structure dictates function. And something else that's really important are that cells are small for not for a reason, right? So going along with this theme, cells are small. This has to contribute to some sort of functional advantage for these cells. So why are cells small? And the reason being is that by having such small surface area to a volume ratio, this is why cells are small. Their volumes are small, but their surface areas are large. This allows for a lot more efficient processes, such as removing waste or getting the nutrients that cells need to survive. So even though cells are very small, some of the cells still have, as I said before, major membrane-bound organelles. Now, we're going to touch upon a lot of these organelles really briefly. So the first one being nucleolus. So the nucleolus is not, it's easy, sometimes mistaken for the nucleus, but it's not the nucleus. It's a tangle of chromatin within the nucleus region that helps to synthesize ribosomal units, so pieces that we need to assemble ribosomes. And this is going to be in the form of something called rRNA. So they're going to be responsible for making rRNA. And the nucleolus is not membrane bound. And this is much like ribosomes, which are, as we know, these tiny little dot structures that make proteins. There are protein factories. And they can be bound on something called the endoplasmic reticulum, or they can be free ribosomes. Now, near the endoplasmic reticulum, there's also something called a peroxisome. Now, you don't need to know too much about this, but just know that what it does is it detoxifies the cell of many of its toxic materials like alcohol. So another toxic compound is hydrogen peroxide, hence the name peroxisome. So it's going to convert hydrogen peroxide. This is a dangerous chemical for the cell, and it's going to convert it into water. So this is going to be really important. Now, the endoplasmic reticulum that I mentioned before is going to be a component that is part of something called the endomembrane system. So the endomembrane system is a a group of organelles, namely the nuclear envelope, so not really an organelle, but the envelope of the nucleus, the endoplasmic reticulum, the Golgi apparatus, and also other vesicle structured organelles, like lysosomes, which are going to be these recycle, trash. You can think of them as disposal organelles that carry a lot of enzymes that break down different compounds in the cell, and also the plasma membrane. So what this system is going to be responsible for is just going to be packaging and also transporting different things either within the cell or to the outside of the cell. Now, the endoplasmic reticulum, there's going to be two types that you need to know. There's the rough and the smooth. In short, the rough is going to be responsible for the synthesis of different proteins on the on the attached ribosomes on the endoplasmic reticulum, and it's also going to store calcium ions. This is something called the sarcoplasmic reticulum, which you don't need to know as of right now. And then last but not least, we have the nucleus. I forgot to mention, just backtracking a bit, the smooth endoplasmic reticulum is going to be responsible for the synthesis of lipids. That's all you need to know for the smooth. And then the nucleus, as we all know, is going to be responsible for DNA replication and also transcription of DNA into mRNA, which is this transcript that is needed to synthesize proteins. Moving on, we have the Golgi apparatus, which is also part of the endomembrane system. Now, this is sometimes also called the Golgi complex, as you see in this photo in orange. And this is essentially a flattened group of membranous sacs that is responsible for processing, packaging, and also secreting different substances, often to outside the cell, so extracellular transport. Lysosomes, we just mentioned that these are going to be sacs that are full of hydrolytic enzymes, and they're going to be responsible for intracellular digestion. And these are going to be found largely in animals only. So while this picture shows a plant cell, know that lysosomes are mainly found in animal cells. Now, plant and animal cells both have, as you know, the next organelle that we have, which is the mitochondria. This is going to be the powerhouse of the cell where the site of cellular respiration occurs, and this is going to be due to this inner membrane that they have called cristae. So again, this idea, moving back to this idea of structure dictating function, the sacs fold on each other to maximize surface area so that the cellular respiration and the processes that occur on the inner membrane can be very efficient. And as we mentioned before, the mitochondria has its own DNA, so this supports the theory of endosymbiosis. And then in this picture, in the middle of the plant cell, we see vacuoles. So this is going to be a large membrane-bound storage organelle responsible for the storage of water and other substances. So in this case, for a plant cell, it's going to be responsible for storing water. This is called a contractile vacuole. Similarly, in plant cells, as we just saw, there's going to be chloroplasts. So those green structures with little pancake-like discs called thylakoids. So like cristae in the mitochondria, thylakoids are these inner membrane sacs that are folded on top of each other that helps to make sugar. They're going to contain a molecule called chlorophyll, which helps to absorb and maximize the absorption of light, which will then convert water and carbon dioxide into glucose. And like mitochondria, they also have their own DNA, again, supporting the theory. And then in both plant and animal cells, they have this cytoskeleton, which is basically this network of different fibers that help the cell to maintain its shape, to anchor, the organelles such as centrosomes and other things that we will learn later. And then it also helps to flow the cytoplasm, so maintaining the flow of cytoplasm, which is really important. Now, the cytoskeleton is going to be composed of many different structures, namely the microtubule, microfilaments, spindle fibers, and also two structures called cilia and flagella, which will be responsible for the motor functions of different cells, such as bacteria, which have which have the flagella structure. And these structures that I mentioned before are largely going to be responsible for moving different things. So microtubules will move different organelles in cellular respiration. Microfilaments will help the cell to change its shape. So this is in the form of actin, this type of protein, and also spindle fibers, which are very important, as we know, in cell replication. The splitting of the cell requires organelles to be distributed equally and the chromosomes to be split. So this is going to be the role of spindle fibers, which will pull the chromosomes apart. And also responsible for this process are structures called centrioles or centrosomes. So two centrioles make up one centrosome. And centrioles are going to be this type of structure that creates these spindle fibers, which will contribute to the pulling apart of chromosomes during cell replication. And in plant cells, they're going to be called microtubule organizing centers, or MTOCs, while in animals, they're going to be called centrosomes. So a little bit of different terminology between the two there. And then last but not least, we have the cell wall, which is going to be this type of structure that's found, as we know, only in plants. And they're actually also going to be found in some types of fungi, but they're mainly found in plants. Sorry for the miscommunication earlier, but in plant cells, they're going to be composed of a large molecule called cellulose, and in fungi, they're going to be made up of chitin. So different types of isomers, molecules there. And then the cell wall is often composed of two different layers. They don't have to be, but there's going to be a primary outer layer in the cell wall, and then also a secondary inner if there's also a secondary layer. And these are going to be largely important to help the plant cell maintain its shape. And then when plant cells touch together, they're going to be glued by this type of substance called middle lamella. And you don't have to know too much about it, but just know that this is what glues the plant cells together. Now, something really important that we have to keep in mind is the plasma membrane. This is going to be extremely important when you're taking the AP biology exam, just because the membrane is going to be such an extensive topic with so many different functions involved that it's going to be critical to know, the different functions and how structure dictates these different types of functions. So as you can see in this picture, there's going to be lots of different structures embedded within the plasma membrane, and there's going to be many things like peripheral proteins, integral proteins that are going to be responsible for a variety of different functions, which we'll talk about right now later on after I explain about this term called selectively permeable and also fluid mosaic model. So these are going to be two very important terms for you to know. So by selectively permeable, we mean that within the plasma membrane, there's going to be these proteins that are embedded in. And these proteins are going to be very specific because of the chemical makeup of these different proteins. So for example, as you can see, there's an integral protein with this little channel in pink in the picture. And the inner core is going to, let's say that in this case, the inner core is very hydrophilic. So this allows the passage of ions in to the channel, while, for example, a small molecule of oxygen can just simply diffuse through, and this is what makes it selectively permeable. There's also going to be a lot of fluidity within this membrane due to the fact that these hydrophobic tails in the phospholipids, as we see in blue in the middle, will help the plasma membrane be very fluid because of the way that those unsaturated fatty tails are very loose. And also cholesterol helps to sort of stabilize that out and also help with stability of the membrane. Now, this membrane is going to have a lot of different functions. Transport, which we mentioned earlier, so the import and export of different molecules into various organelles or into the cell. There's also going to be enzymatic activity, so these different proteins, many of them could be enzymes that help to catalyze different reactions. There's going to be signal transduction, which we'll touch upon later. Cell-to-cell recognition, so glycolipids and glycoproteins are, as we see in this picture, these little structures, kind of Y-shaped, they're green, and they're going to be very important in cell-to-cell recognition. So glycolipids are these carbs with lipids attached, and then glycoproteins are going to be these structures that are a hybrid between a carbohydrate molecule and a type of protein. So this is going to be really important. And then the cell-to-cell attachments are not to be confused with cell-to-cell recognition, but cell-to-cell attachments are going to be various structures like desmosomes that you don't have to know at this point because these are very deep and in detail. But for now, just know that cell-to-cell attachments and also attachments to the cytoskeleton, as we touched upon earlier, and the extracellular matrix. So these different proteins within the extracellular matrix, such as collagen, are also going to be done through this plasma membrane. Moving on, we have cell transport. So this is often also going to include the cell membrane of the cell, and sometimes also the membrane of organelles. And there's going to be two types, but the first type is going to be something called passive transport. And this is going to be when molecules are transported from a high concentration to a low concentration. So when we have two sides, as we see in the picture, there's going to be one side with a higher concentration to the side with lower concentration, and we call this moving with the gradient. And there can be two main types. We have simple diffusion and facilitated diffusion. So in simple diffusion, molecules that are often very small, such as oxygen, can just directly diffuse through the plasma membrane just because they're small enough. And in contrast, we have larger molecules, let's say glucose, that needs to be imported into the cell. And if molecules like glucose are big and need to be transported, they are done with the help of transport proteins, as we see in the picture. So these are two types. There's also osmosis, which is a fancy term for saying the passive transport of water molecules from a higher to lower concentration gradient. And then there's also some terminology that's extremely important that involves passive transport. So we have solvent and solute. So solvent is going to be the thing that dissolves a substance, which is referred to as a solute. So for example, water, we know this is the universal solvent. There could be, let's say, sodium chloride, table salt that you want to dissolve. That's going to be the solute. And then three terms, we have hypertonic, hypotonic, and also isotonic. So hypertonic is when relative to a concentration, let's say that you have on one side, and another side is more has a higher concentration of a certain molecule. We would say that the other side is hypertonic to our side. Conversely, if we have the other side being a lower concentration, we would say that that side is hypotonic to our concentration. And if the other side is equal to our side, the concentrations are equal, we would label that as isotonic. And also water potential, just another term that just means the ability of water molecules to move from a higher to lower concentration. Now, the other type of cell transport is active transport, which involves carriers or pumps. So a very important type of pump or carrier is a very important type of pump is the sodium-potassium pump, which is important for maintaining the concentrations of these two different ions across a cell, which is very important for nerve cells to conduct that action potential. And this process is the most important thing that you have to know about this process is that it requires energy. So if we see on the picture on the right, there's going to be a molecule called ATP, which we all know is the molecule that helps to provide energy for many cellular functions that we need. And this is produced during cellular respiration, and it's extremely important. One of the reasons is for transport. Now, this is also really important in the electron transport chain, also in cellular respiration, and also vesicle transport. So, apologies for this typo, but vesicle transport, such as endocytosis, so the bringing in of different molecules through vesicles, or exo, so bringing out, is also going to require this this active transport. And then just another term called bulk flow. You don't need to know this in depth, but just know that this refers to the movement of liquid from a source. So where the molecule that you want to transport comes from to the sink, to the destination. And two good examples are blood in animals and also sap in plants. Moving on, we have cell communication. This is a very important idea that requires a lot of the structures that we learned before. So the cytoskeleton has various structures. The plasma membrane also does, and this is going to be important, as we said, for cell-to-cell signaling. In bacteria, there's going to be something called quorum sensing, and this is going to be just know that this is a term to refer to communication between bacteria cells, so the horizontal transfer of genes, and also often bioluminescence, so the signal of light from one bacteria cell to another. There's also direct contact. As we said before, there's various structures that allow for cell-to-cell communication, and two examples are gap junctions in heart cells. You don't have to know these, or plasmodesmata. These structures that are also really important for cell transport. And this is going to allow for passage of materials directly from cell to cell. There's also local signaling, which is like in nerve cells, just the transport of different molecules from a short distance. So growth factors, neurotransmitters between nerve cells, this is going to be local signaling. And then there's long distance signaling, which we know as the transport of hormones in the bloodstream, so from one gland to another. And this often requires receptors, which is also found on the surface of the plasma membrane. Now, there's 3 different stages of cell signaling. So, as I mentioned before, once a molecule comes into contact with a receptor, so that molecule is referred to as a ligand. this is going to initiate the reception stage, which is the first stage. Now, when the ligand binds to the surface receptor, it's going to trigger a response. These receptors can be on the surface or they can be within the cell. And once these receptors are bound to the ligand, this brings about a response that the cell can then convert into a response. And this process of converting that signal to a response is called transduction. And the response often comes in the form of transcription or also sometimes cytoplasmic action. So kind of like squeezing the cell, helping it move, or rearranging different microfilaments or different structures within the cell. And then our last concept for today is apoptosis. Just know that this is another term for programmed cell death, and it requires signaling pathways once again. So as I said before, the plasma membrane is extremely important for relaying different signals. One of the functions being apoptosis. This is going to play a major role in development. So in embryonic development, our fingers have when we are in the womb, our fingers actually have this layer, kind of like a web structure, this layer of skin attaching our two fingers together. But apoptosis is actually responsible for giving us these gaps in between our fingers so that we don't have webbed hands. And also, apoptosis, just know that it also has a very important function in maintaining homeostasis. So when cells, for example, have damaged DNA or perhaps there's misfolded proteins, that's going to signal to the cell that something's wrong and that we need to immediately take action so that the cell that has these damaged structures dies, or it's like kind of like cell suicide in a sense, so that so that no damage occurs to the cell. So thanks for watching this video. This was a lot of content, but very important nonetheless. Please subscribe for more content and like, share, and comment if you have any questions or concerns. So this is it for the third video, the cell, and we'll see you next time.